Jennifer, thank you for organizing this meeting and thank you for inviting me. I like it that we can see what we're saying there. So I'm just going to read my talk over from what's showing up there so I don't have to memorize it. So this talk will be a bit more computational than some of the others yesterday and today. I hope that's okay. And as always, yeah, please interrupt as we go along. My lab has, one of the questions my lab has always been interested in is how the brain tells time and decodes temporal information. And whenever somebody says that, it's important to remember that that's not a particularly informative sentence because the brain tells time over many, many orders of magnitude and uses fundamentally different mechanisms to tell time across these orders of magnitude. So it's very important to define what time scale I'm going to be interested in. And I'm going to really be in the time scale here of hundreds of milliseconds to a few seconds. And this is the time scale in many ways. That's probably the most sophisticated form of temporal processing the brain performs. So music, of course, is a beautiful example. If I just ask you to discriminate a 1/8 note from a 1/16 note, somehow your brain is doing a little simple temporal discrimination task, a simple temporal computation that allows you to make that distinction. But you're also telling time right now as I talk. You're paying attention to the intervals and pauses and durations of the phonemes coming out of my mouth. And a common example of that is if I say two sentences such as, they gave her cat food or they gave her cat food. So in both of those cases, you have two very fundamental different meanings based on the pauses, the prosody of my speech. So your brain is continuously telling time inevitably on that time scale. So it's important to define that in addition to those examples that I just gave you, sensory timing, the brain is also doing motor timing on that time scale. When you're anticipating a yellow light or playing a piano or even double clicking on a mouse, you have to appropriately time your motor actions, or of course in complex motor coordination such as in sports. Now, I'm going to focus on timing, sensory timing here, because the brain really does have fundamental different mechanisms to cope with these different temporal problems that the brain performs. So an example of sensory timing that we've studied for many years, even when I was at UCSF, is just a simple sensory discrimination task. And this is an interval discrimination task in which you'd be sitting at a computer and I would ask you, I don't know if we have sound here. Probably not, but that's okay. We don't know if you can hear that. Yeah, we'll add that here. But a simple auditory discrimination task in which you'd just be given, it's okay, simple, and you'd be asked, was the first interval shorter or longer? And you'd say that the first interval was, the second interval was longer in that case. And then that was 100 milliseconds versus 150 milliseconds. And then we'd make it more difficult based on your performance and adaptive protocol to measure your threshold. And if we train people on that at an interval, standard interval of 100 milliseconds, people get better over time. This is a form of temporal perceptual learning, which is not trivial. This takes time. It takes like 5 to 10 days to have that type of improvement. What's interesting here is whatever the neural mechanisms underlying temporal processing, they're improving. But those mechanisms seem to be specific to the interval that you're trained on. So if we get the pre-post values at 100 milliseconds before training and after training, and this is the trained interval, you get better. But at the other intervals, 50, 200, 500, people do not improve. So this tells us, this constrains a bit the potential neural mechanisms that are underlying timing at the neural level. Now, the two hypotheses I'm going to talk about today are first, that short-term synaptic plasticity is responsible or partly responsible for sensory timing on this scale. This is an old story that is mostly published work, and that's probably not new to many of you. The second story, which is mostly unpublished, part of this hypothesis is that STP, short-term synaptic plasticity, which I will define in a moment because this generates a lot of confusion, undergoes long-term plasticity. So we're going to refer to this as long-term plasticity of short-term plasticity. This is in keeping with one of the themes that Jennifer had in mind in organizing this sort of cross-temporal interaction of neural mechanisms. So what do I mean by short-term synaptic plasticity? That used, I think Tom referred to that, I think Cliff referred to that. So I'm going to carefully define this because people really do mean different things by short-term synaptic plasticity. I'm defining my use of the term, which I think is the most common in the literature as it was described going all the way back to Eccles in 1942, as a use-dependent changes in synaptic strength generally happening on the sub-second scale. This is totally non-NMDA dependent and totally non-associative. Okay, so people use this term differently. This is absolutely robust. There's one of the most robust forms of synaptic plasticity in the brain. Almost all synapses have short-term synaptic plasticity. That short-term synaptic plasticity can take various forms depending on the synapse. It can take form of short-term depression or short-term facilitation. And normally this form of short-term synaptic plasticity has been seen as a bit hardwired, I would say. Short-term synaptic plasticity exists at almost all the synapses, all the different synapse classes, and we often define different types of short-term synaptic plasticity at different synapse classes as a signature of those classes. So one of the most famous here, for example, is the excitatory connections on the somatostatin neurons. These are generally shown to be quite facilitatory. So you get this very robust short-term facilitation in general. The excitatory to PV neurons tends to have mild or moderate depression, as do the IPSPs from PV to excitatory neurons. The IPSPs from somatostatin to PV neurons tends to be sort of stable as well. But there's a lot of variability. So for example, in the pyramidal neurons, excitatory to excitatory, it can vary a lot between mild facilitation to sort of almost steady state to pretty significant depression. And these short forms of short-term plasticity, they're dependent on many factors, not only the cell types, but the developmental age, the state of the system. Allison, for example, has shown that during upstates or in the presence of GABA agonists, it can change the short-term plasticity between the excitatory neurons. From depressing to facilitating, for example. Now, a key point that I'd like to highlight is to immediately bring this into the interval discrimination task I mentioned. If we sit down and just sort of make up a circuit, hardwire circuit, we can make interval selective neurons using short-term synaptic plasticity. So let me just go through that. I forgot to start my own timer. I'm not very good with timing here. If we just have a simple disynaptic circuit, okay, so you have an input. We can think of this as a thalamocortical input to the cortex, perhaps, and it's activating an excitatory neuron and an inhibitory neuron, and an inhibitory neuron in turn activates or inhibits the excitatory neuron. And each one of these has whatever signature of short-term plasticity we're assuming, based often on experimental data. So in this little circuit, if we play our cards right, we can see that we can create a 100 millisecond detector. That means if you give 2 pulses at 50 milliseconds, the excitatory neuron will not fire. Why? Well, because you have facilitation at the synapse, but the facilitation is vetoed by the facilitation in the inhibitory neuron. Now, at 100 milliseconds, this fires here because of facilitation that on that synapse that was also present here, but now the facilitation to the inhibitory neuron wasn't enough to fire the inhibitory neuron here, so you didn't veto that. And for the longer interval, the facilitation was not sufficiently robust or long-lasting to fire the neuron here. So the point is that if we play a little hardwired game here of tuning those synapses, we can create interval selectivity. What we're showing here is that even in this simple circuit, you can temporally tune the neuron to be 50 millisecond selective, or 100 millisecond selective in green, or 200 millisecond selective, by changing the appropriate synaptic weights now. So STP is constant, but by just tuning the weights, so this is the input to the excitatory, this is the input to the inhibitory neuron, by tuning the weights here, you can shift here. And for example, if you make all the weights strong, you get white, which is the blue plus the green plus the red. So here you have non-selective responses. So depending on the tuning of both the short-term synaptic plasticity and the inhibitory and the total synaptic strength, you can create your interval selective neurons. Now, LTP, short-term plasticity, and LTP do interact in some cases. So here's an old example of a bit of short-term depression on a layer 2-3 pyramidal neuron, and you can see it undergoes depression. Short-term depression, short-term synaptic depression. Then we can induce LTP by pairing presynaptic activity with postsynaptic activity. So this is the pairing process. So you're seeing LTP online develop. And what you're seeing here is the black here is the baseline, the red is at the appropriate minute mark here, and you can see LTP developing. What you see, though, after, is that really you had your standard LTP, but the short-term plasticity interacted in a manner that the last pulses were not potentiated. So the LTP in this case was specific to the first pulses here. So there's this strong interaction. In some cases, and this will probably generate a bit of confusion that I should come back to. Now, and this is, I think, came up again maybe in one of Peter Diane's questions yesterday. Even that protocol, that same protocol in the hippocampus and neocortex, they both generate LTP, but they have fundamental differences in terms of their effect on the interaction of STP and LTP. So when we induce LTP in the neocortex, you get this interaction where the first one is potentiated more than the last. And in CA1, you generally get this very uniform potentiation, which is just basically scaling the responses. This is consistent with a purely postsynaptic mechanism. I think Rob might have some data here where you showed some interactions between LTP and STP in one of your papers. And I think that's a pretty robust finding. Yeah, Manish. No. So here, a multiplicative factor, meaning like the postsynaptic amount of postsynaptic AMPA receptors, would absolutely explain it. But here, if it was just a multiplicative factor, for the later pulses? Here, the potentiation. So here what you should don't, yeah, let's not confuse the temporal summation with the EPSP amplitude. So here the EPSP amplitude is the same here and here, but it's riding on previous that were stronger here. So yeah, no, it can't be just multiplicative in that case. Okay, now this is in a very simple network. So what we and others have done is proposed in general under the name of something that we called state-dependent networks. Nowadays, people refer to those as reservoir networks. And the idea here is any complex dynamical system can be used in principle to decode time. And if you have a liquid or a pond here, and I showed you these little raindrops falling, with a bit of math, you can easily tell me which raindrop fell first, and that it was this raindrop and then that raindrop. And you could even calculate the interval because it's a dynamical system, and the radius of these ripples are expanding at a certain rate, and you could figure out the interval between those. So the idea here is that in a neural network, the state of the system, just like the state of the liquid there, that we define as, say, the diameter or the radius of the ripples or the amplitude of the ripples, has information about the past, right? It has information about elapsed time. So in the brain, we can try to look at the system in a simple neocortical circuit and ask, can we use the dynamical changes in the neural network to decode time or encode time? Now, normally, when we talk about the state of a neural network, we're referring to which neurons are active. But there's also what we're going to call the hidden state. In the hidden state of the network, we have all these other properties that many people have already talked about today, like just short-term synaptic plasticity and MDA receptors, GABA B currents, and slow ionic conductances, and maybe astrocytes, as Cliff mentioned earlier today. So these are both dynamical systems. These are both changing in time, whether you're looking at the neurons that are currently active or whatever the time constants, the biochemical processes that are evolving in time that will change the behavior of the network. So if we go back to that interval discrimination task and we have our hypothetical auditory cortex circuit here, and we have a bunch of excitatory neurons and inhibitory neurons, before the two tones, the beep beep, let's say the network is inactive. In response to the first tone over here, some excitatory neurons will respond and some inhibitory neurons will respond. Then, right before the second tone, in this example, the network is silent again. So the active state is the same, but the hidden state has to be different, right? So there's still information about what's happened in the past here that's different from here. So some synapses are facilitated, some neurons, some synapses are inhibited, depressed and facilitated. So that means that when the same input comes in, let's say this is the same exact input here and here, the effect on the network has to be different. So when you ping the network, if you will, because the synapse strength is different here and here, the responses should be different. And the active responses here should tell you information about the past, meaning the interval. And in that case, when you have that type of simulation, and you look at the response to the 100 millisecond, so 1, 2, or the 200 millisecond, 1, 2, those two images are overlaid. So of course, each one of this is the voltage of a simulated neuron. And we can go in here and find neurons that respond to 100 milliseconds or to 200 milliseconds. So this is the standard idea of sort of this complex network, just because of its complexity, just because of its high dimensionality, will generate useful responses that you can use to decode something. And this is sort of the reservoir aspect of that. Yeah, Peter.
Not the 100 milliseconds. If you double the time between each process, then the hidden state would have evolved in the same way. So therefore, you would vary it.
Right. Are you asking how far out you can go?
No, how variable. So if you change that, so if you change the macroscopic gap between the constants, you would presumably expect a certain degradation of the quality of the.
Yeah, absolutely.
So you can make it shorter or make it longer.
Yeah. So maybe I think you're getting a bit to the temporal resolution. And because these time constants, they're dynamics, the equations that control them, the derivative, if you will, tends to get flatter as time goes by. Their ability to decode time gets worse and worse. So at some point you can't do it, and I'll come back to that. But that's exactly what happens psychophysically as well. So as a percentage, your threshold at 100 milliseconds might be 10%, might be plus or minus 10, but at 200 milliseconds, your threshold is not plus or minus 10, it's more or less. Yeah, okay. Yeah, okay. I think the next slide might help answer that question, which is a really good one. So the strengths of this type of model is that the state of the network encodes not only the present stimulus, but the past stimuli. The weakness of the model is that the state of the network encodes not only the present stimulus, but the past stimuli, right? So, and sometimes as I'm speaking to you, if I say onto or into, to has fundamentally different meanings, right? Because it's interpreted, everything I'm saying for the most part is interpreted in the context of what happened. It's state dependent. There's nothing more natural in our brains, right? Each word I say is interpreted in the context of the previous. But other times that's bad. If I'm saying 4-5-2 or 6-3-2, you want that one to be the same, right? Because a 2 is a 2. So there's advantages and disadvantages. So prediction that arises, and maybe this is relevant to Peter's question, is because it's the hidden state, you can't reset short-term plasticity at will, right? It's not like you could potentially reset the active state of the network at will by inhibiting everything at once. So this is sort of, I think, what maybe Peter was asking. So if we look at the state of the system, now importantly, when I'm plotting the state of the system here in PCA space, this is not simply the active neurons. This is the state of the synapses, whether they're going to be facilitated or inhibited. So it's very high dimensional space, right? If you have 100 neurons and they're fully connected, you have 10,000 synapses. So this this state can be defined in 10,000 dimensional space. But it's sort of low dimensional because the equations are all the same. So if we just have this here, and we have our T0 here, and it goes through time. I'm sorry, this is a mess, it's hard to follow, but the bottom line is it starts here and comes back here. If we give another stimulus right here before it had time to reset, then the prediction is timing will be difficult. But if we give it enough time to reset, then we predict, well, then it's possible to do that task that I just gave you and compare the first interval with the second interval. And so we had actually tested that. And the test was, sorry, I don't think we're listening to this. Well, maybe you can hear that. So there's a short interval, what we call a short ISI. And if I asked you which was the first shorter or longer, I think that's a bit harder. Here, it should be a bit easier. Oops. So it's easier to parse, right? And sure enough, the thresholds change significantly. Now, you might say, well, of course, it's hard to do things that happen quick. A nice control that's possible to do here is we do the same interval, interstimulus interval, but now at different frequencies, because now it's a different network. You don't have to reset that pond because the auditory cortex is tonotopically organized. So if I ask you to do this one, so now you're clearly using different tones. And it turns out that your ability to do that is better than your ability to do that, presumed because they're not interfering. Was that sort of your question, Pete? Okay. Okay, now, and this idea, by the way, of short-term synaptic plasticity being used to do timing has now been used in other systems. This is a model of cerebellum by David de Gregorio that came out, I think, last year or this year. Yeah, sorry.
You said because auditory cortex is small topic. In a lot of other auditory conditions, there's one topic also. But is there any reason why interval discrimination would be cortex dependent? Or can animals indicate cortex?
Yeah. So there's actually a study by Carlos Brody that suggests that you can do interval discrimination without auditory cortex. That does so, and I think here I would agree with Allison, that doesn't mean you're not normally using your auditory to do this task, but because there's many of these stations. Now, we would make the prediction that we'd still get that same result, but it would be maybe it would be less dramatic or something, so I don't know. Yeah?
If you look at the other studies for learning, when they train people on race discrimination or interval discrimination, you find transfer from auditory training to visual training, but obviously not vice versa. And there's evidence that some of this is not local.
So let me just clarify this. So there is reasonable transfer at that given interval to other modalities in some cases. So when you learn the 100 millisecond interval, you can transfer to other. You don't transfer, and generally almost never transfer to different intervals. So our interpretation of the intermodality transfer is that what you're doing is creating a better representation of the target. And indeed, there's good data by Beverly Wright that transfer takes many days. So the initial learning is quick, the intramodal, but the intramodal, but the intermodal takes additional days. So it's implying that there is indeed some sort of higher level process going on there. Okay, now I want to introduce the concept of long-term plasticity of short-term plasticity. And this is the more novel part, and this is addressing this sort of cross-modal interactions. So typically, short-term plasticity is viewed as a more or less hardwired property or an epiphenomena of synaptic strength. If the synapses are strong, maybe you have depression. If the synapses are weak, maybe you have facilitation. What we're going to propose is that the temporal profile of short-term plasticity is itself learned. So normally, if you have a depressing synapse or a facilitating synapse, there's a standard Sawdix-Markram model that we can use to simulate that has a depressing factor and a facilitating factor, and that gives us enough degrees of freedom to almost capture any type of short-term plasticity you happen to measure in your cells. And it's defined by these taus, this time constant for depression, time constant for facilitation, and this U value, which is seen as sort of the percentage of available transmitter that's released. It's a bit like release probability, but not exactly. So playing with these parameters, the tau d, tau f, tau, you can go from one to the other. So what we're suggesting here is that the parameters in red are learned, or might be learned, through some sort of metaplasticity. And so people have talked about Hebb, pre-post pairing, STDP, depending on the timing here. So what we're suggesting here is that in addition to the standard Hebbian story, a synapse might learn to exhibit facilitation or depression depending on when the pairing happened. So if you have a bunch of presynaptic trains, action potentials, and you pair the late synapses, maybe you're teaching the neuron or the synapse that it wants to have facilitation, because that's when the postsynaptic neuron will fire. If you pair early, that means the hypothesis is that you would be teaching the synapse, or computation, from a computational perspective, it would make sense for that synapse to have depression, because it wants to causally fire the postsynaptic cell early, as opposed to late. So the idea here is that you would be training, there's a learning rule in place to train short-term synaptic plasticity. We had shown a while back, we developed a rule based on that, that when you incorporate this long-term plasticity, short-term plasticity with a fairly simple learning rule, and you train a hypothetical simulated net neuron to discriminate Poisson patterns, that the presence of long-term plasticity, of short-term plasticity, this temporal synaptic plasticity here, significantly improves performance. So this is errors here, whereas in the absence of STP or fixed STP, performance is significantly worse. Okay, now I'm going to shift a bit to a bit away from the neuroscience as to an approach in artificial neural networks or machine learning. So is this hypothetical form of plasticity a way that the brain or artificial neural networks can enhance their ability to do temporal tasks? So
hypothetical. If you've been looking at changes in temporal ratios after LTV forever, even Sawdix and Markram have their redistribution.
Yeah.
So it's sort of a generalization of all of that, in other words, or how do you?
Two things. So yeah, I mean, that's that slide I already showed you of our own data, right? So there is an interaction in neocortex. Now, in general, those protocols don't address what we're saying because you're just always pairing at the beginning. So what we need to do is just pair at specific intervals. But more importantly, Rob, I think, so you actually have some interesting data that speaks to that. I think it's in your 2006, 2007 paper with STDP, where you played around with whether it was the fourth pulse or was fifth pair. But I don't think you looked at short-term plasticity of those results there, which always frustrated me, Rob. But yeah. But either way, we've done a bit of that, and I really think this requires protein synthesis. So I really don't think you're going to see it within a half hour, is my actual answer to that. And I can go back to that. I think I have some data to suggest that. Okay, so here's, people probably know, and I'm sort of losing track of time here. Everybody's familiar with the importance of the XOR problem, or some people may not be familiar with the XOR problem. So the XOR problem is a simple problem in spatial, in just sort of standard artificial neural networks, that if you want to train a postsynaptic neuron to respond to this, there's two inputs, input 1 and input 2. It shouldn't respond to this, it should respond to this, it should respond to this, but not to this. And mathematically, it can be shown, well, that's impossible if you just have a linear system. And this is what we're going to call the spatial temporal XOR problem. So in the spatial temporal XOR problem, you have two inputs, each fires twice, and you don't want it to respond if the input 2 goes on twice, input 1 goes on twice, but only to the second stimulus if 1, 2 or 2, 1, or vice versa. So the only way to solve that we can show here is if this network has short-term plasticity, not only short-term plasticity, but long-term plasticity or short-term plasticity. So we can then train the network to solve this task using now a gradient descent. So now we're using a totally non-biological approach in which we're using gradient descent to train those tau f's, tau d's, and u values. Okay, so this was just a proof of principle that first it computationally enhances the power of the model, and second that you can use gradient descent to train those values. And one of the tasks we did to test this, oh, let me just say something here. So how do machine learning models, artificial neural networks generally deal with time? So generally, I think this is a weakness or at least a disconnect between the biology and machine learning. So in one of the standard approaches in deep neural network models, they spatialize time. So this is totally artificial, in which you have basically a retina for t-3, a retina for t-2, and a retina for t-1. You have different inputs for each point in time, and so you're buffering time. That's what I'm going to refer to the spatialization of time, which is obviously not biological. A lot of models use recurrent networks. We've done this, and a lot of people have done, and that's, of course, more biological, in which the information is reverberating about the past within the recurrent network. So information about T3 is somehow still present in the network at T because it's reverberating in there. Transformers don't also do something incredibly non-biological. Transformers are like GPT-4, in which they have this clever, actually very clever mechanism of each moment in time is labeled with a timestamp, if you will, of its order in, for example, a sentence. So T3, T2, T1 has this sort they call positional encoding, in which you basically have a timestamp that's overlaid on your stimulus. So what we're proposing here is that another way to go about this is with short-term synaptic plasticity, because short-term synaptic plasticity contains information about the past in the actual synapses without having to invoke these additional mechanisms. So Morse code has always been my favorite example of how good we are at temporal information processing. So it's amazing people who are good at Morse code. Again, when they just and they say, oh, that was this word, and it's just like one jumble of words and beep, beep, beeps. But so here we're doing a neural network simulation, and we're training a simple neural network, just a feed-forward neural network, no recurrency, in which the synapses have STP, and that STP is trainable. So those variables, and we can compare the performance and the ability to learn all the letters in Morse code. So this is the inputs, these are, this is the hidden layer, those are the inputs, these are the outputs. It's a bit hard to see here, but you have the specific activation of A, B, C, and so forth here. And this is the overall performance, and if you have plastic STP, performance is significantly better than with fixed STP or without STP. And this is true, by the way, even when you control for the degrees of freedom, because obviously when you're training STP, you have more trainable variables. Okay, so I'm just going to go through one or two more slides here, Jennifer. And so we'd like to test this hypothesis now experimentally. And we're working on this. I'm not going to talk about much of that today. But one way we wanted to get an insight into this was with the following question. If you're recording from a trio of neurons, for example, and this is your presynaptic neuron, and you have two postsynaptic neurons, is the STP correlated at this synapse and this synapse? Because STP is presynaptic, so I should have said that early on. So I think a lot of people would predict that if these are the same classes, that the STP should be correlated at this synapse or in this synapse. So is that cell-specific STP? Or is STP synapse-specific, meaning at this synapse you can have facilitation, at this synapse you can have depression. So we used the data set from the Allen Institute, in which they recorded from 1,371 pairs. And there were not that many pairs of triplets from excitatory neurons to two excitatory neurons. but we got the pairs that they had and looked at the correlation. We fit the STP here and here to the classic STP model, and we asked if those parameters are correlated in the postsynaptic partners that share the same presynaptic neuron, if that's clear enough. And what we find there is that the correlation is really very poor. So the U value, the probability of release, the tau Fs and the tau Ds, the correlations of those synapses that share the same presynaptic neuron is basically, they're basically not correlated. So to us, our interpretation, this was just a first step towards our prediction, is that this is an indication that maybe each synapse does learn independently of the other synapse the short-term plasticity parameters. Yeah?
How does the learner fit in general to the data? So when you fit the results spider model, the synapses.
The fits to the data are very good because it's over parameterized, if you will. So the Markram Todix model fits almost all the data very, very well. The problem is that, you know, it almost overfits in that there's many, it's degenerate. So you have to be a bit careful. Okay, let me summarize here. So the first is a simple observation that STP is observed in all neocortical synapses, but there's relatively little focus on the computational function of STP, and even less so on whether STP is learned. That is, are there synaptic learning rules that govern the temporal profiles of STP? And that's one of the things that we'll be focusing on in the future. So we propose that STP injects short-term memory into the hidden state of neural circuits, allowing them to process temporal patterns on the sub-second time scale. So it's not a coincidence that STP, the time constants are a few hundred milliseconds, and it's not a coincidence that my speech and music has time constants on a few hundred milliseconds. So the brain evolved to use those inherent mechanisms to do the task, sensory processing task that we're all doing right now. And lastly, we also propose that STP may not be hardwired, that is genetically determined, property of synapses, but learned in an experience-dependent manner, thus enhancing the ability of neocortical circuits to process temporal information. Thanks to some people in my lab, mostly Shun Lin here, Uma Carmarker, and Tiago Carvalho. Thank you.
So we're gonna take questions specifically for Dean until about 1:15, and then we'll switch to the larger discussions. You can continue asking questions of Dean and of the other speakers as well, okay? So, let's see.
Thanks so much. Really enjoyed the talk. So have you tried your LPSP rule in the context of your reservoir? So you showed it in a sort of simpler domain where you only have this feedforward network. Once you're in the reservoir, then the complexities of how sort of the chaotic dynamics evolve maybe quite.
This is basically that. So here we do have the LPSP, and I should be clear, it's not like there's the LPSP rule, there's number, and this has one of those types of LPSP rule. So that, yes, so this is basically a reservoir network plus the LPSP.
So how big is the network? Like how many neurons? How chaotic is the network?
I'm sorry. Here, this is just the one, and then we have done the other case in which you have, I don't think it's that big, maybe 100 neurons. But part of the idea in that case is that so reservoir networks are sort of poorly defined, as you probably know. So in the standard reservoir network, you have the recurrency, and in the first models that were proposed, that we published and Wolfgang Maas published, there was also STP. In the later models, they removed STP, and then you run into the problem in which you absolutely need the recurrency, and then you get into this chaotic regime that generates these instabilities. But here you can avoid that chaotic regime by having these longer time constants.
Thank you.
Sorry. So a reservoir network is just an idea that even a randomly connected network has computational power. So not all networks have to be trained. Just if you wire up a network in a reasonable regime and then you give it input, it will generate high-dimensional representations of that input that can be used to do computation. So the analogy of the liquid was a good one. And people actually do this as a toy model, where you can throw in different patterns of activity into a liquid and then discriminate those temporal patterns on the liquid. So
The question is whether in this reservoir network you can also
training, yeah.
train by changing STP with your channel.
Yeah, might no longer be called the reservoir network, but anyways, yeah.
So, Dean, maybe following on that question, if you look at the recent reanalysis of the Allen Institute data, that's also compatible with just totally random expression of mechanisms for STP across the network, right? So if the network is large enough, you already have in the reservoir all the timescales you need to represent anything.
That's one hypothesis, yeah.
So in that situation, then the role of plasticity would not be to be able to store the new patterns. It would be to protect yourself against instabilities. Is that how it would work?
I'm sorry, I see. Yes, the reservoir network has the potential to do complex computations, but it doesn't mean it can do any computation or do those computations efficiently. So the idea is that, and this is my view about the brain, by the way, this is my view about the sensory cortex, is that it is initially randomly connected, and it is functioning as a reservoir, but then it's perfected on top of that through learning mechanisms to further optimize the problems that you have to. So my brain might be naively a reservoir network when I'm young and I don't know if I'm going to have to learn Mandarin or English, but obviously then through training, it has these long-term plasticity mechanisms. Yeah.
So I always think about like short-term plasticity, how that translates. Sorry, thank you for the talk, by the way. So in terms of short-term plasticity, I'm always wondering how that translates to the in vivo case, where you have like spontaneous activity and constant synaptic barrages, right? And when people have measured thalamocortical synapses in vivo, they're like constantly depressed. And so do you have any thoughts about that? And do your kind of network simulations include spontaneous background activity?
They can. It might not be enough to your satisfaction, but there's many, it's an important question. So not only do you have spontaneous activity, it turns out temperature and neuromodulators are a big thing too. So we know those change short-term plasticity, and so that would have to be incorporated in the model. What I would argue, what I would love to do, and again, I mean, so I think a prediction that would arise, and this again relates to a recent paper by Rob, the Schiavo paper, in which the prediction is that the animals learn something new, a new temporal thing, in his case, mouse calls, that the STP would be changing precisely to learn those. So then you could go in those slices and specifically address the short-term plasticity specifically, which was, we can talk about. But yeah, but it's an important question, Christine.
Jen?
Yeah.
Oh, sorry. Okay.
Yeah.
Very interesting talk. Thank you. My impression was that if you go to a single synapse level, then short-term plasticity is reflected in the probability of release, right?
It's correlated with,
right.
It's not totally determined by the probabilities, but it's correlated with probability.
Right. So,
and even that, you have to be careful with that statement, but yeah, continue.
Okay, so the pair part pulse facilitation is actually the increase of the probability of release to the second spike, right? For example.
Not only.
Okay.
Because sometimes it's just, you can have release on both synapses, but more release on the second. You can have, so if you have pulse 1 and pulse 2, you can have release on both,
So
but
different quantal size.
Yes, or different number of vessels.
Okay.
Yeah.
But then I thought there was also a relationship between the probability of release on a synapse and whether it is depressing or facilitating, so that the low probability of release synapses are likely to be facilitating and the high probability of release synapses are depressing.
Yeah.
Wouldn't that go along with your LTP of STP story, or that's not?
I'm
relevant?
sorry. Yeah, I have the answer to that here.
Okay.
So that is the more or less standard story. I think it's a bit too simplified because on a case-by-case basis, you can find situations in which the release probability is the same, or the initial EPSP amplitude is the same, but one has facilitation, one has depression. So in the Allen data, this sort of addresses your question, Mitra. Here we're correlating first amplitude of the EPSP, which is sort of a reasonable measure of probability release. And with the different values, we can just look up here. Here's the strongest correlation. So there's a reasonable correlation between when the first amplitude is stronger, that this is going, up that way.
But this is multisynaptic, no?
This is pairs of neurons, which could have more than one bouton.
Exactly.
That's a big unknown. So
exactly.
It's hard to get around that one. But
right.
yeah.
But that's why I went to the probability
Yeah.
measurement, because then you have a scale.
Yeah. So that's hard to get to that. But remember, you also need to take into account the tau F and the tau D. So presumably, and this is really cool, neurons seem to have these calcium sensors in place that may control the tau Fs and the tau Ds in addition to the probability release. So we're suggesting those tau Fs and tau Ds are also learned.
Sorry, but just tau Fs is the maximum STP?
Tau Fs determine the temporal profile, how long the short-term facilitation lasts, or how long it takes to recover from depression.
Between the initial and the second spike, or between the pairs?
Between the first and the second spike, or through the dynamics if you have a bunch of presynaptic spikes. Yeah. So those three parameters allow you to do everything. Yeah.
So I'm considering Christine's comment, and I think it's a big complication here. But the question that I had for you is, because we know something about how these short-term plasticity dynamics decay over time, you might predict that there are certain intervals that are easier for us to learn and other intervals that are really hard for us to learn. Do you see that in psychophysics? Do you see that in your simulations? And I guess that one question there, there may be so much hysteresis because of so much spontaneous activity that you really don't get to see things like that, because synapses that dynamic range is broadened by their prior experience. But perhaps you could silence the network for a while, kind of reset everything, and then see that, we only, 150 milliseconds is the best interval, and, other intervals are really hard.
I don't think we see that. I mean, temporal perceptual learning is defined sort of between tens of milliseconds to half a second. Above that, it's sort of different mechanisms because you start having conscious perception, conscious rehearsal of the interval. But if I give you a 1/8, 1/16 note, it's too quick for any higher order processing, and same thing with Morse code. Now, within that range, I don't really think there's...
Wait, I think I need to understand that better. You're saying a 1/8 note and a 1/16 note.
Yeah, so that's like...
I can't tell you the difference between them?
No, no, you absolutely can. But it's too quick for you to perceive that as duration consciously. It's sort of just purely perceptual. It's like almost reflexive. So that's my only point. Whereas if I give you a 1 second versus a 2 second, you start counting in your head. That's my point. So there are different mechanisms. We know that for what we call automatic timing and reflective timing. Now, to your question of preferred intervals, no, I don't think there's any indication that there's preferred intervals. And what we would argue is that it's learned, right? So if I give you an interval that you have to learn because your language or your music background is such that 100 milliseconds are very important, or your Morse code is very important, that would change the time constant. So we would like to look at that within those slices.
But why shouldn't there be intervals that are easiest to learn?
No, that's not by the models, not really, because remember, these are all smooth curves, right? Okay. Because they have smooth derivatives. And
so you have such a diversity of short-term plasticity because of that spontaneous activity? I mean, maybe it's created by that background of...
I think it's just the diversity. I mean, in that simple circuit that I showed you, with only 3 neurons, then your hands are a bit more tied, right? So you don't have as much flex. So there you have preferred intervals you could have in a simple circuit like this. But as soon as this becomes bigger, no, I don't think so. Because of the inherent variability of the system and the richness of the system, which we could call a reservoir, it has the sufficient flexibility to account for all the intervals. Yeah, fair enough.
Is it the case that associatively the typical distribution obeys some kind of Weber law?
Absolutely, absolutely.
About naturally or not naturally?
It does, and I think this was related to the slowing of the derivatives. So all these things have a derivative that slows down, and I think the Weber fraction comes in because you lose the precision because the change in those state space programs.
Where's the slowing of the derivatives? Is it information that is?
Yeah, well, I mean, you can sort of see it here. If I look at the derivative of the change in any STP curve, it changes quickly, and then the amplitude slows down with a tau. I mean, in the equations, it's just an exponential decay almost, basically. So the derivative is slowing down.
Okay, great. Let's move on to the broader discussion.